Coxiella burnettii is a short, gram-negative rod that causes a disease called Q fever. This bacteria is highly resistant to environmental stressors, including high temperatures and ultraviolet light, and spreads to humans from mammals like cows, so Q fever is considered a zoonotic infection. Now, Coxiella burnettii is gram-negative, meaning that its cell wall has a thin peptidoglycan layer, so it cannot retain a stain called crystal violet stain. Instead, it stains pink with safranin dye used during gram staining, so it looks like a little pink rod under the microscope. C. burnettii is also non-motile, and it can undergo endosporulation when it feels threatened by the environment, like when the temperature becomes too high or too low, in case of extreme dryness, or when there's harmful radiation around. Endosporulation describes a process in which the bacteria starts by replicating its DNA. Then, it forms a wall inside the cell, isolating a portion from the rest of the cell. Let's call it the mother cell. Next, the plasma membrane of the cell surrounds the mother cell and then pinches it off, forming a separate body known as a forespore. The forespore then invaginates into the mother cell and gets completely engulfed by it. Inside the dying mother cell, the forespore loses water and accumulates calcium, and at the same time gets wrapped in a super tough cortex from the dying mother cell. At this point, the endospore is able to resist heat, harsh chemicals, digestive enzymes, and even antibiotics. Finally, as the mother cell dies off, the endospore is released outside. Surprisingly, an endospore can last over 1,000 years, waiting for favorable conditions to come and then germinate into the bacterial or vegetative form, which can then grow, divide, and infect organisms. Coxiella burnettii is also an obligate intracellular organism because it can't make its own energy in the form of ATP, so it can only replicate inside other cells like our macrophages. The bacteria does not routinely grow on standard agar plates, but can be grown using animal inoculation or can be cultivated in embryonated eggs or cell culture. Now, the primary disease that Coxiella burnettii causes is called Q fever, an atypical pneumonia. This disease results when people inhale the Coxiella spores. This usually happens when spores are blown downwind from animal farms or when they come into close contact with livestock that are harboring the bacteria. So people at high risk for infections include people in contact with farm animals, particularly cattle or the amniotic fluid of cattle, and slaughterhouse workers. In terms of pathogenesis, Coxiella burnettii is transmitted through aerosolized spores. However, the exact mechanism of disease is poorly understood. What is known is that C. burnettii has a capsule that expresses proteins called phase 1 and phase 2 antigens that allow it to infect cells and replicate. When C. burnettii expresses a protein called phase 1 antigen, it is highly infectious, and a single bacterium can be enough to cause acute disease in humans. Phase 1 antigen binds to receptors on phagocytes like macrophages in the lung. Normally, phagocytes destroy invading bacteria via endocytosis, which is when bacteria is engulfed by the phagocyte, wrapped in a vesicles called a phagosome. The phagosome would normally merge with another intracellular organelle called a lysosome, forming a phagolysosome. Lysosomes release hydrolytic enzymes into the phagolysosome, which normally destroy the invading bacteria. However, C. burnettii can escape the immune recognition by using type 4 secretion system, or T4SS for short, which is a collection of proteins that can dampen the immune response. So macrophages don't gobble up C. burnettii like they should. What's more, if they do eat the bacteria, C. burnettii use another virulence factor on its outer membrane called non-endotoxin lipopolysaccharide, or LPS for short. LPS inhibits the fusion between the phagosome and the lysosome, allowing C. burnettii to avoid intracellular death. So once C. burnettii brucella is safe from intracellular destruction, T4SS starts secreting effector molecules. There's a whole bunch of these molecules, and their exact role is not yet well understood. But what we do know is that they help the bacteria replicate in large numbers. Once C. burnettii starts to replicate, it begins to express phase 2 antigen on its capsule. which marks the transition from acute to chronic disease. Now, the ability to survive and replicate inside macrophages also makes it able to travel through the bloodstream to multiple organs, such as lymph nodes, liver, and spleen. Additionally, when the macrophage becomes too small for the growing number of C. burnettii, it bursts, releasing the bacteria into the bloodstream. And from there, the bacteria can spread to other organs, such as bone marrow, central nervous system, bones, heart, liver, and lungs. It can take anywhere from 2 to 3 weeks to develop acute symptoms, which are generally nonspecific and include high fevers, chills, sweating, headache, fatigue, muscle aches, a non-productive cough, nausea, vomiting, or diarrhea. Importantly, Coxiella does not cause a rash. Symptoms of chronic infections include chronic cough, frequent headaches, and intermittent fevers. Chronic disease can rarely cause a complication called infective endocarditis, or an infection of the inner lining of the heart chambers and heart valves. This complication can happen to people with normal or native heart tissue, although people with prosthetic or damaged valves are at higher risk. This can lead to cardiac murmurs, septic emboli, arrhythmias, chest pain, and shortness of breath. The diagnosis of Q fever is tricky because the disease presents with nonspecific signs and symptoms. Laboratory studies usually demonstrate an elevated white blood cell count, elevated inflammatory markers like ESR or CRP, elevated liver enzymes if the bacteria invade the liver, or positive blood cultures if the bacteria make their way into the bloodstream. The diagnosis should be suspected on a history of frequent contact with animals like cattle and subsequent development of symptoms. Serologic testing against phase 1 or phase 2 antigens is used to confirm the disease. PCR can also be done to directly identify bacterial DNA. Another method to confirm diagnosis is the Weil-Felix test. This is where a blood sample gets mixed with antigens from another bacteria called Proteus. A positive test for Coxiella burnettii is when agglutination or clumping does not occur. Conversely, a negative test results when agglutination or clumping occurs when a person's blood gets mixed with Proteus antigens. Treatment of Q fever involves doxycycline for 14 days for most patients with acute Q fever, and subsequent serologic testing at 3 and 6 months after the treatment to confirm resolution of the infection. Alright, as a quick recap, Coxiella burnettii, also referred to as Q fever, is a short, pleomorphic, gram-negative rod that is a strict intracellular bacterium. It is a zoonotic infection that is transmitted via aerosolized spores. It causes atypical pneumonia in humans, as well as a host of other nonspecific signs and symptoms. Treatment involves a course of doxycycline.